Plant-based compounds, particularly polyphenol like resveratrol and curcumin, have captured growing scientific interest for their potential to influence aging at the cellular level, specifically through their effects on telomeres, inflammation, oxidative stress, and DNA repair pathways. These natural bioactives, found abundantly in certain herbs, spices, and fruits, do more than act as antioxidants. They interact with key longevity-associated genes and enzymes, suggesting they may actively preserve telomere length and enhance genomic stability over time. Resveratrol, a stilbenoid found in the skin of red grapes, berries, and peanuts, is perhaps one of the most well-researched polyphenol in the context of aging. One of its most intriguing mechanisms of action is the activation of sirtuin proteins, especially SIRT1, a family of NAD+ dependent enzymes that regulate metabolic efficiency, stress resistance, mitochondrial function, and DNA repair. Sirtuins are thought to be central regulators of healthy aging, and there is evidence that their activation can influence telomerase activity, the enzyme responsible for elongating telomeres in certain cells. By upregulating SIRT1, resveratrol may help stabilize telomeric DNA and prevent premature cellular senescence. Although most of this research is based on animal models or in vitro studies, the implications for human health and longevity are compelling, especially as these pathways are evolutionarily conserved. In addition to its effect on telomeres, resveratrol also has broad anti-inflammatory and antioxidative effects, which are both highly relevant to cellular aging. Chronic low-grade inflammation and elevated oxidative stress are among the most destructive forces to telomere integrity. Resveratrol suppresses pro-inflammatory cytokines such as TNF-alpha and IL-6 and enhances antioxidant enzyme systems including catalase and superoxide dismutase. These systemic effects contribute to a cellular environment that is more conducive to DNA preservation, telomere maintenance, and delayed onset of senescence. Curcumin, the active polyphenolic component of turmeric, is another standout plant compound with powerful anti-aging potential. Known for its potent anti-inflammatory properties, curcumin inhibits nuclear factor kappa B, NF-B, a transcription factor that regulates many genes involved in inflammation and immune response. Through this mechanism, curcumin reduces the burden of oxidative and inflammatory damage that accelerates telomere attrition. In vitro studies have demonstrated curcumin's ability to preserve telomere length in fibroblasts and reduce cellular aging markers, possibly through modulation of telomerase activity and oxidative defense pathways. Beyond inflammation and oxidation, curcumin also supports mitochondrial health and autophagy, two critical processes in slowing down cellular aging. Healthy mitochondria generate less free radical damage and maintain optimal energy production for cell repair and division, while autophagy helps clear damaged cellular components that could otherwise trigger stress signals, contributing to telomere erosion. This multi-layered cellular maintenance effect makes curcumin a particularly valuable candidate in the landscape of nutraceuticals aimed at promoting longevity. Importantly, these plant-based compounds often work better in the context of a synergistic diet rather than in isolated supplement form. When consumed as part of a diverse, polyphenol-rich dietary pattern, such as the Mediterranean or Asian-style plant-based diet, their bioavailability and systemic effects are enhanced through interactions with fiber, fats, and other micronutrients. For instance, curcumin absorption increases significantly when combined with black pepper, piperine, or healthy fats like olive oil, while resveratrol's effects may be supported by other polyphenol like quercetin. While more long-term human trials are needed to definitively establish their telomere-preserving effects, early-phase research on both resveratrol and curcumin strongly supports their role as molecular protectors. They influence not just symptoms of aging, but root-level regulators of cellular health, offering a non-invasive, natural approach to supporting DNA repair, immune modulation, and genomic stability. These compounds, taken regularly as part of a health-conscious lifestyle, appear to tip the balance toward resilience, regeneration, and prolonged cellular function. Ultimately, the potential of plant-based compounds to preserve telomeres exemplifies how nature itself may offer some of the most elegant solutions to the challenge of aging. Rather than working against the body with aggressive or artificial interventions, compounds like resveratrol and curcumin support the body's own repair mechanisms, activating ancient biological systems that evolved to maintain life. In doing so, they don't just add years to life, they may help add quality to those years, right down to the DNA.